Welcome to an edition of CCSD Living Environment. Today we're going to wrap up talking about cell transport. So cell transport part 2, if you will. The last time we talked about passive transport and the types thereof. No energy required by the cell. You're moving with the concentration gradient. Essentially you're moving downhill. Movement from high to low. So let's change gears and talk about active transport. This is where we require the use of energy or ATP from the cell. Active transport. ATP. Usually moves molecules from a low concentration to a high concentration, so we're going against the concentration gradient. You're trying to ride that bike uphill. You have to invest energy to make that happen. Here I have just a few molecules inside the cell, low concentration, a ton of molecules outside the cell, high concentration, and I'm going to try to push these guys out. That's going to require a lot of energy. There's already a lot here, and now you're trying to push more into that. That's not passive transport. That's very active. It's requiring the cell a lot of energy to make that happen. An example of that inside the human body would be with the sodium-potassium pump involved with your nervous system and allowing nervous function to work, which we'll get to more of that later, but it does happen even though you look at it and say, boy, it doesn't seem like the cells would want to invest that kind of energy. Every now and then we need to, and when it comes to nerve conduction, it's a good thing we do. What I'd like you to do is turn back in your note packet to the very beginning. And this picture is listed in the very beginning of your notes. So we've got the high concentration at the top of the hill, low concentration at the bottom of the hill. If we are riding down the hill, no energy, that is passive transport. Examples being diffusion, osmosis, facilitated diffusion, no energy is invested on this side. If I go from low concentration to high concentration, I'm trying to ride that bike up the hill. I need to invest a ton of energy, a lot of ATP to make that happen. That is active transport. Energy is involved, energy is being used. Active transport. We want to go against the concentration gradient. Please go back to the beginning of your note packet and make sure that you add this information to the picture that's there. A couple definitions that we need to talk about, and they're involved with the transport within cells. They're not necessarily passive transport or active transport, but they are things that take place within a cell. The first one being what we call bulk transport. With bulk transport, it's a transport of large molecules packaged in a membrane-bound sac that we move across the cell membrane. So it's a large molecule, and basically you package it up and you kind of trick the cell to let you move it in. So they call that bulk transport. Bulk being buying in large quantity. For example, if you go to Sam's Club or BJ's or Costco, they sell things in bulk. They sell large quantities of it at a cheaper price. So with bulk transport, we're transporting a larger molecule into the cell. And then finally, there's two other terms that we need to look at. The first one being exocytosis. Exo, remember, means out. Cyto means cell. So we're moving something out of the cell. Waste packaged by the Golgi body fuses with the cell membrane, and they're secreted out of the cell. The opposite of that would be endocytosis. Endo meaning in, cyto meaning cell, so we're going to move into the cell. The cell membrane surrounds the desired macromolecule and moves them into the cell. Macro means large. So the cell membrane of the cell surrounds this big particle and then pulls it into the cell. There's two different types of endocytosis. The first type of endocytosis is called phagocytosis. The entire cell is engulfed and brought into the cell. And the other example is called pinocytosis. It's a fluid, basically a droplet that's taken in. And in your note packet, it gives you a picture to show you. And you can see on the one where you're surrounding it with the cell membrane and bringing that larger particle in, it's showing you phagocytosis or endocytosis. And then it's showing you, okay, we want to get this out of the cell, bring it to the cell membrane and release it. And that would be exocytosis. So that pretty much finishes up the different types of cell transport. and kind of takes care of what is it that the cell membrane is doing. Remember, all of this stuff is about the cell membrane and its job as far as allowing things into or out of the cell. And in some cases, like moving large particles in, you kind of have to trick the cell membrane. Because remember, what is the most important characteristic that the cell membrane uses to control whether something can move in or out? And yes, hopefully you just said size. So that finishes up our bit on cell transport. I will see you soon.